Okay, so I think we can make a start. So welcome to the Palaeoverse Lecture Series, everyone. This lecture series is focused on software development and methodological approaches in paleontology, with the aim of promoting the use of new tools in the field and to give researchers a platform to showcase their work. Now, with this series, we hope to shake up the usual type of lecture by having a mixed format with time dedicated to the presentation, demonstration, and discussion of the software or method presented. Now, typically these sessions will run for about 50 minutes in total, and we'll run this series throughout the year. And it's generally on the last Thursday of each month, unless they overlap with holidays, such as this week. So if you're interested in presenting a new method or software you've been developing or implementing, please feel free to reach out. We're always kind of happy to hear from the community and looking for speakers. But before moving on, just a few quick introductions today. My name is Lewis Jones, and with me today I have Erin Dillon, Will Gertie, and Harriet Drage. The four of us are on the core team of Palaeoverse, an organization aiming to bring the paleobiological community together to share resources, reach agreed standards, and improve reproducibility in the field. Now, if you want to know more about Palaeoverse, please check out our website at palaeoverse.org, and I believe the link's just being dropped in the chat. And you can also check out our GitHub organization. The link again is being dropped in the chat. But today, that's all from me. We'll now give you some helpful tips and instructions for this webinar, and Erin will introduce this month's speaker. So thank you very much, and please enjoy the lecture. Over to you, Will.
Thanks, Lewis. As Lewis mentioned, the format of the seminar will consist of a talk by our speaker, followed by about 10 to 15 minutes at the end dedicated to question and answer and discussion. My fellow hosts and I will help field your questions and moderate this discussion. Please note that this seminar is being recorded and will be made available on the Palaeoverse YouTube channel after it ends today. However, we'll stop the recording before the question and answer period in case you'd prefer not to be recorded when asking your questions. Before we get started, I'd like to point out a couple of useful Zoom controls at the bottom of your screen. First, you'll see a Q&A button. You can use this to submit questions to our speaker. During the question and answer session, we'll then read out your question or invite you to turn on your microphone and ask it directly. There's also a raise hand button. Alternatively, during the question and answer, you can raise your hand and we'll call on you and invite you to turn on your microphone so you can ask your question. There's also a chat feature, which is currently only enabled for you to chat with us, the hosts, in case you have any technical difficulties, but will allow you to chat with other attendees during the question and answer period. There's also a button for closed captions, and you can turn that on and view the transcript at any time. Last, I'd like to highlight a couple of ground rules. Our aim is to cultivate an open, respectful, and inclusive environment during this lecture series where we can learn from each other. With this in mind, we ask that you please be respectful and kind when engaging with the speaker and fellow attendees, particularly when asking questions and interacting with others in the chat. Any form of harassment or inappropriate behavior will be grounds for removal from the webinar platform. And for more information, please refer to the Palaeoverse Code of Conduct, which can be found on our website and is being placed in the chat right now. With that, I'll pass it over to Erin to introduce today's speaker.
Wonderful. So thanks, Lewis and Will, for getting us all up to speed on how the lecture series works. Today I have the great honor of introducing our speaker, Dr. Carrie Tyler. So Dr. Tyler is a marine conservation paleobiologist and a professor at the University of Nevada, Las Vegas in the Department of Geoscience. Carrie's research focuses on understanding the evolution of marine ecosystems, critically how food webs respond to episodes of environmental change, and the relationship between biodiversity and ecosystem dynamics through time. Her work also seeks to assess the quality of the fossil record and determine ways to use this taphonomic information to inform paleoecology and conservation. And I believe Carrie will be sharing more about this particular avenue of research today and some of its implications for understanding and measuring functional diversity in the fossil record. So without further ado, we're very excited to hear from Dr. Carrie Tyler with a talk titled The High Fidelity of Trophic Structure in Ancient Marine Ecosystems, From Functional Diversity to Food Webs. Please join me in welcoming Dr. Tyler to our virtual stage.
Thank you so much, Erin, and thank you all for hosting this fabulous event. As I said earlier, I've really enjoyed this lecture series. I'm going to talk about the reliability of particularly trophic structure and how it's preserved in the fossil record, and with a focus on functional diversity. And one of the reasons for that is, if I can click through my slides. There we go. Is because functional diversity has become really a sort of a gold standard metric in conservation for measuring ecosystem health. And there's been this big switch from concentrating on the number of species in an ecosystem and conserving number of species to conserving functions. And so here on this figure on the left, you can see is an example of why. In the bottom panel, I can see that there's fairly high species diversity. There's some sort of disturbance or perturbation here where species diversity drops. And then after that disturbance, the number of species returns to a number that's comparable to prior to the disturbance. But in that same ecosystem, while functional diversity is quite high before the disturbance and drops, functional diversity doesn't recover, even though the number of species does. And this is something called functional homogenization, which we see quite often in disturbed ecosystems today. Um, One issue with functional diversity is that there's not a one-to-one relationship between the number of functions and the number of species, which is what you see clearly here in the bottom left. And this relationship varies, we think, by environment, perhaps latitudinally. It's not quite well understood yet. But you can see here we have these different points of different colors for tropical temperate environments in the Galapagos here. And you can clearly see this, here's the linear relationship here, is not a one-to-one, and the slope of that line is different in different places. So it would be really helpful to understand how functional diversity works, what its relationship is to number of species, because that would really shed some light on the best approaches for conservation today and maintaining and preserving healthy ecosystems or restoring them. Here's another global map. This is fish diversity. Here you can see the number of species on this global map around coasts, particularly with cold colors indicating low diversity and warm colors high. And you can see on the panel on the right that functional diversity doesn't match that pattern of species diversity. There are some areas that do have higher functional diversity where we have high species diversity, but again, it's not automatic. So, for example, here in this region, we can see there's very high species diversity, but very low functional diversity, for example. So we can't assume that because we have a lot of taxa, we also have a lot of functions. And so really what happens when an ecosystem is disturbed today, and what happens when we lose biodiversity as well. We really need to understand these relationships. And so one of the ways I explore and try to answer these questions is to look for emergent properties in ecosystems. And a great place to do that, of course, is the fossil record. So how do we, there's some big questions in ecology. How do we explain how ecosystems persist? How do they self-organize? What are the rules? Again, what are the emergent properties that arise from the attributes of the ecosystem? So again, the structure and the functioning, which of course in turn arise from interactions. And really it's because we need to know those rules if we want to preserve and manage ecosystems today and to prioritize funding, which as we all know is restricted. And again, a great place to do this is the fossil record. We have, all of space and time to play with, as I say to my students. Here's the classic Sapkowski curve. We can see we've had these changes in the number of taxa through time that are very well known. So we can look at the fossil record and look at times when we've had changes in the number of taxa, and also when we've had big dramatic changes in ecosystems. And try to understand again these emergent properties and how ecosystems really work. So we have several events in the fossil record that are great times to ask these kinds of questions and explore how ecosystems work. I've highlighted here three really major events that are really well known that are sort of all characterized as revolutions in some way, particularly ecological revolutions. And so these trends in increasing taxonomic diversity and other changes in ecosystems have sort of led to this assumption that ecosystem structure and functioning has changed through time, and that ecosystems have become more ecologically complex. So here's another great example. We're going to use this approach quite a lot today. These are cubes that represent ecospace. This is a hypothetical ecospace. This approach was put forward by Andy Bush and Dick Bombach. And it's sort of a useful way to think about how organisms are occupying ecospace through time. So here we have Ediacaran through recent, a few intervals in between. And so the filled cubes here represent ecologies that have taxa in those ecologies. So sort of a filling of ecospace through time. And this has really influenced, I think, how we think about ecosystems in paleoecology, that these cubes are becoming fuller through time, essentially, right? That there's more and more ecologies have arisen through time. But if you look at food web structure, there is some work that has suggested that even though we might have more taxa, that in fact ecosystem structure hasn't changed from the Cambrian to today. So this works from Jennifer Dunn and her group, where they compared Cambrian food webs with modern food webs, and they concluded that there haven't been any big structural changes, and that they're really comparable in terms of their diversity and ecological diversity and complexity. So really, this isn't a given that ecosystems have become more complex or ecologically diverse through time. So this is something I'm really interested in exploring. And these methods, both functional diversity and food webs, are widely used to ask a lot of different kinds of questions as well, by a lot of people in my group included. So here are just some other examples of work using functions, which are sort of a core input for construction of food webs, or analyzing functional diversity directly. So even though this is Bombachian cubes are ecological ecospace, they are actually the same as functional diversity, and they're using the same traits to measure functional diversity. So these methods are really important. And so we sort of want to understand how reliable they are in the fossil record if we're going to use them in these ways. So the way we measure functional diversity is using traits of organisms. So for example, here we have a swimming crab. I know it's a swimming crab because one of its traits is that it has these swimming paddles on its back legs here. So that's a trait. And this is the sort of classic, again, Bombachian cube, where we can take those traits to characterize this organism. So for example, this crab is pelagic. It's largely predatory, and it's fully motile and fast. It's a swimmer. And so this filled in cube here, that's its functional, those are its functional traits, the combination of these three. You can also get more complicated, use more than 3 traits, of course, and instead of being constrained to these cubes, the more typical way to measure functional diversity in ecology is using multivariate approaches. Here's the convex hulls for 2 functional spaces in a multivariate analysis. And as I said, we also actually use this for food webs to construct a food web. We use these traits as well to characterize the relationships and to create these interactions between organisms as well. We use both traits in both of those ways. So constructing the food web also relies on these traits and on functional diversity. So here is an example of how we construct a food web. You need a list of taxa, of course, to start with for your community. And then we organize those taxa into guilds or groups. You might also refer to these as trophic aggregations. This is the way it's described in modern ecosystems. You use trophic aggregation to group things together that have the same ecologies and do things in the same ways and have the same predators and prey. And again, we use these traits to do this. Here, this is the method that myself and my collaborators use when we construct food webs. We group things into guilds. Guilds is a common term and a common approach in paleoecology. But just as a reminder, a guild is a group of species potentially using the same resources in similar ways. And for our food webs, they share other properties, such as their life habit, their range of body sizes, and also their predators. And so this is what we use to construct the relationships. We also call this a meta network. There's a lot of food web jargon. And this is our input for our analyses. So we can compare functional structure from there using that meta network. We can model species level food webs, which is what we usually do to ask questions about the food web. And then we can use those models to test and ask questions about ecosystem dynamics. So again, functional diversity and traits are essentially the input for food web construction. So here's sort of visually what I mean by trophic aggregation. These boxes are essentially our guilds. Again, this is just trophically aggregating individual species together that have the same ecologies and the same groups of predators and prey, which are highlighted here with the colors. There's another approach that's been fairly widely used to construct food webs, and this relies even more on those functional traits. Here, the Bombachian traits, motility, tearing, and feeding, are the input for that directly for that construction of the food webs, and they're the main input with the addition of body size, which is also a functional trait. So functional traits are a huge component of constructing this food web. So if these aren't reliable, we can't ask those questions with food webs, and it would, of course, we'd have to think about how we interpret food webs through geologic time and in the fossil record in particular. So this is the question that I'm interested in. Really big picture, can we trust the fossil record as a source of historical data? And I say historical data here because I also, of course, think that this is a valuable source of data for conservation, right? So conservation paleobiology. We know there's differences. We know that death assemblages and the fossil record suffer from several biases and processes that alter the fossil record. So we have temporal mixing, we have transport, disarticulation, and decay, differential preservation, and differences in relative abundance, right? We know all of these things are happening, but how do they affect the ecology, right, when we actually try to compare ecosystems? So I asked these questions and tried to test this using a classic approach. It's a live dead study from the coast of North Carolina. And so what we did was a series of dredges that are onshore, offshore transects. And we also tried to capture a few other types of environments here. So every point on this map is a dredge location. And in each of these dredge locations, we have at least 3 dredges with various types of equipment, including a Van Veen grab, just to try to get the very shallow subsurface. And many of these sites were sampled in more than 1 year. So we went back to this location over and over again, several years. So some of these sites have many years of data. And this is what we pull up in the dredge, and we have, of course, live and dead mixed together. We take a bulk sample of the dead, and then we would go through the sample and survey all of the live organisms at each of these sites. And then we take the death assemblage back to the lab to process, and then that way we'd have a sympatric comparison of the live community and the accumulating death assemblage at every one of these points. This is a multi-taxic study, so I'm again working on food webs. I'm interested in whole communities and everything in the ecosystem. So every single type of invertebrate was included, with the exception of encrusters. And so here you can see some of that variety. Here's a whip coral, sea urchin, moon snail. We have a crab. So this includes everything that we were able to identify. So again, we have these dredge surveys. We kind of identified all of those invertebrates, and all of them were, of course, vouchered and photographed, and assembled this live dead data set for comparison. This is macrofauna only. Identifying species on a boat, if you've done that, is quite challenging, and you're not able to identify microfossils on a moving or microorganisms on a moving boat quite easily. We were, sampling a lot of sites as well, so we had to move fairly quickly. So this is macrofauna, and the other reason to do that is because that's what we tend to use in, again, paleoecology, food web reconstruction, things like that I'm working on. And so we wanted this to be applicable to this broad range of work, which generally uses macrofauna. Almost all of the taxa are identified 2 species. There were a few encrusters. We excluded them from the data set. It's just difficult to consistently identify those. And we used a 5mm mesh for the death assemblage, again, to make this comparable to what we would pick out with the naked eye on the boat. So that we're comparing apples to apples here with our live survey and our death assemblage. We also converted number of skeletal components to number of individuals. So everything I'm showing you here is based on number of individuals in the death assemblage. Some of those conversions are easy. A crab only has 2 kilo, for example. How many spines are on an urchin? That's a philosophical question almost. So we tried to very conservatively estimate to make those adjustments. So again, here we're comparing numbers of individuals, not numbers of components. So our live assemblage, we had about 179 species and a little over 11,500 individuals. Death assemblage was 160 species and a little over 57,100 individuals. And again, I want to point out that to get 57,000 individuals, that's more than 90,000 skeletal components. And so this is a very, very large and robust data set. So let's first look at these functional traits in the sort of classic Bombachian style. Again, these are widely used in paleoecology. So for every species in the data set assigned, these 3 traits actually assigned more. I'll show you that later in the talk. But these traits are readily available from places like polytraits, EOL, worm, sea life base, etc. And of course, the paleobiology database has also a great record of these traits for marine invertebrates, which is why these 3 traits are very well used in paleoecology. So again, tearing, feeding, and motility. This is to the cube with everything sort of together. Usually you see it this way, where there's sort of a space between each of those tiers. So it's the same cube, essentially. So here's the comparison between the live on the left and the dead on the right for the number of individuals. So this is abundance data, again, with those Bombachian cubes. So we go from deep and faunal to pelagic. Here we have feeding, and here we have motility. And so the cool colors here indicate fewer individuals. The warm colors indicate many individuals. This is a log scale. But what you can see is that the similar cubes are filled in both of these. And cubes that have a lot of individuals in the live tend to have a lot of individuals in the dead. And of course, we have some accumulations of individuals in some groups. This very dark red cube here, which is only light pink in the live, is of course has more individuals because that's the functional entity or functional group for mollusks. So that's not surprising that there'd be more individuals in that group. But I think more interesting is to compare richness, right? The number of taxa. So here we have the live, the dead, and also created a hypothetical fossil record here. So the fossil data here are only based on any species in our data set that had a fossil record or a fossil was recorded somewhere and documented in the paleobiology database. So this is a hypothetical fossil record based on our actual data. And again, we can only do this for a number of species, not abundance, because we don't have abundance data for that. But for, again, the live, the dead, and even the fossil, we see the same cubes are generally filled. Of course, we have this accumulation of richness based on mollusks, which do preserve very well. But generally, these colors are fairly consistent. And this match is really, really good. Here, if we look at each of those traits individually, we can see, so here's the death assemblage and the live assemblage, again, this number of taxa. And here on the right, we have the fossil assemblage. And you can see for each of those functional groups, there's a really strong relationship between the live and the dead. And here, the shaded regions show the confidence intervals, so for 99 to 50%, again, based on our bootstrapping here with 10,000 iterations. So you see this is a really, really strong relationship here with the richness is highly congruent between the live and the dead and the live and the fossil as well. This is another way to test that relationship. This is a Spearman rank correlation. I'm only showing you the top 20 functions here. But they're all green because we have a strong congruence here again between the live and the dead. It's a fairly strong relationship here between the rank order of the number of individuals in each of these functional groups between the live and the dead. So again, it's a very reliable relationship here. Um, so again, there's a lot of factors that seem to inform the distribution of or inform ecosystem functioning. The diversity or the types of functions seem to be really important in ecosystem health. again, for conservation. Redundancy is really important for ecosystems. That's the number of species divided by the number of functions. That's how we calculate that, right? So essentially insurance. Do you have extra species in that functional group? We can also look at how vulnerable functional groups are, right? So if you lose species, are you going to lose a function? This is quantified by looking at the percentage of functions with only one species. And we can also look at sort of extra insurance over redundancy, and that's measured as the percentage of species that fill functions over the mean redundancy. So essentially, these are all different ways to quantify functional diversity. And these are measures that are used to quantify functional diversity in modern ecology and in the fossil record as well. So I'm just going to look at a couple of those metrics and how that stacks up. Again, this is just our live, dead, and fossil for all three of these. Here we can see the number of species. Not surprisingly, there's more taxa in the death assemblage and even the fossil relative to the live, but it's not a huge increase. In terms of having insurance and redundancy within functions, there is an increase in the death assemblage and the fossil relative to the live. And the same for vulnerability. There's more vulnerable functions, or a greater percentage of functions are vulnerable in the dead and the fossil, whereas over redundancy increases. And as you notice, these other relationships, the fossil is a little lower than the dead, and this is the one exception where the fossil was higher than the dead. This is what the curve looks like if you rank the functional entities. This is the number of species per functional entity here. It's just sort of smoothed out instead of being a histogram. So they're ranked the same for all of these. And this is a good way to sort of visualize the functional diversity. This arrow here is showing how many functions are vulnerable. So the functional vulnerability here in the fossil assemblage is 41%. In the death assemblage, it's 42%. So these are the functions that are vulnerable. And you can see it's only 18% in the live. This dashed line here represents the redundancy. So all of the functions that are above this line are over redundant. And again, you see it's quite consistent across all of these. So we have a bigger peak here in terms of number of species in the highest ranked functional entity. So the shape of this is a little different. But it's still fairly comparable across all of these. I'm not going to go too much into it, but this gray line here just represents if taxa were randomly distributed amongst these functional entities, what would this look like based on 1,000 iterations? And you can see in pretty much most of these functions, they differ from what a random assignment would look like. So let's talk about a more sophisticated method of and way of quantifying functional diversity. This is multivariate approaches to measuring functional diversity are much more common. And in this case, I took it one step further. So not just using multivariate functional diversity measures, but doing so with fuzzy coding. So in fuzzy coding, we're able to take those traits. And I also used many more traits. So we used 8 instead of just the 3 classic Bombachian traits. And I also was able to consider that many organisms don't just do one thing, right? So in the sort of classical approach, you have to pick one feeding method, for example, one life habit for an organism. This crab, though, doesn't just do one thing. This particular species of crab, and in fact, the other one I showed you as well, has more than one feeding type. So this crab is an opportunistic scavenger and an ambushed predator. So if I just called it a predator, it'd actually be missing some of its ecology. And with fuzzy coding, I can account for that. So here is one trait. Let's say that this is feeding type. And I can assign a heavier weight and say that most of the time it's an ambush predator, but some of the time it's an opportunistic scavenger. And so we can have this fuzzy approach where we consider that organisms don't necessarily only do one thing or have one thing for some of these categories. And so that's called fuzzy coding. So we use this fuzzy coding approach to look at this, and then you can do your multivariate analysis of functional space. So here's the most straightforward measure here, which is a measure of functional richness, again, using the fuzzy coded approach with more traits. And functional richness here is measured as the proportion of a functional space used. It's essentially the volume of the convex hull. Here's our live, dead, and fossil again. And here's the convex hull for the live, and I've mapped it onto all 3 of these so you can compare how they differ from the live assemblage. And I've also colored the different phyla in this data set. So here we have mollusks. There were a few individuals of brachiopods in the live. We have annelids and cnidaria, arthropods, and of course echinoderms. And so those are marked which parts of the functional space those phyla occupy in each of these. And you can see that the volume of that functional space is maintained fairly well from the dead, in the dead and the fossil from the live. We're really only missing this little bit sliver here and here of that functional space. Most of it is still there and is still recorded by the death assemblage and the fossil assemblage. So this is really robust. We then took that a step further and looked at how that might vary with environment. We did have some other data from another paper on this same data set where we observed that there's this depth gradient in this ecosystem that is, well, again, we've documented that fairly well. And so we're able to break out these environments based on that relationship that we'd observed previously and documented previously. So we characterized all of these sites as offshore, again, based on their depth, and near shore here. That's how we made that division, was again with the depth. And then we also have a few back barrier sites here, meaning that they're behind these barrier islands. These regions have higher salinity and of course are protected from open ocean waves. So this is a slightly different environment. And so we were able to compare these environments and look at how this changes. So here's that further offshore or deeper sites, and near shore, shallower sites, and those back barrier sites. And again, you can see that from the live to the dead to the fossil, in each of these environments is still fairly well maintained. That functional space is well represented. And the volume of that convex hull is pretty much there as we move from live to fossil in all three of those environments. So this is really good news. So whether you're focused in on one environment for your data, or you're looking at a broader data set, for example, across a shelf, which is usually what I do, it's reliable either way. And this, of course, perhaps because these methods are relying on whether or not a function has species in it, so it's perhaps not surprising. There's lots of other ways to measure and quantify functional diversity with these multivariate metrics. So I showed you functional richness again, which is the volume of that convex hull. But there's a lot of other ways to do it. I'm just going to show you the results from two others very briefly: functional divergence and functional dispersion. Divergence is looking at the deviation from the centroid, so how far away are the traits, how spread out are they in the functional space. And then dispersion is the deviation of the values from the center. So here, again, we're comparing the distances essentially in the pathways. And again, for all of these metrics, for divergence and dispersion, we again see it's very consistent from the live to the dead to the fossil. And you can still see that missing space there in the white just to show you for comparison. But here are the actual values at the top of each of these, and they're very similar. So 0.87, 0.87, 0.86, right? Very, very comparable. If you were to measure, that's divergence, and here's dispersion, 0.85. 0.84, 0.84, right? So very, very similar measures for each of those. And here's when the same analysis, but we've added in abundance data. So you can also use abundance data in these analyses. And here I've just added functional richness back again, but divergence and dispersion. And again, they're very, very similar for the live and the dead. 0.78 versus 0.7, for example. So you can also include your abundance data if you have it, and it still will work really well. Although this, when you add abundance in, it does tend to get a little more different than just presence absence. So that might be harder to use. So this is one place, right? One case study. But I also looked at a survey of the literature and modern studies of functional diversity from, again, benthic macroinvertebrates, which is what we were looking at. I looked at papers from every paper I could find from every different type of environment. So from polar environments all the way to tropical, and looked at the faunal composition, right, of their studies. Again, these are from benthic ecosystems, and in almost all of the studies, so 22 out of the 27 studies that reported the number of individuals per phylum or class, those studies all had the same top three organisms that we found in this study. So mollusk, arthropods, and annelids are the three most diverse and abundant macrobenthic groups in the majority of studies of benthic macroinvertebrates. And so in terms of how generalizable are these results, I think they are fairly generalizable because it's the same groups that we're using here in this study. And again, in a whole suite of environments, it's still these same groups that are the most that are making up most of the organisms in these studies of functional diversity. So, in terms of how reliable is the fossil record, and I refer to this as ecological fidelity, we saw that it's quite high. Many ecological metrics are comparable, and that's despite differences in the relative proportions of species and the richness of different phyla and classes in the death assemblages. So it's not that they're not different in terms of their composition; it's that those differences don't lead to different answers when you're quantifying ecology. I again didn't show that here, but coming out of this same data set, we've looked at environmental gradients and spatial relationships. They're all preserved in the death assemblage. And in this case here, I showed you the functional fidelity for benthic invertebrates, and it's very high. And so this gives me a lot of confidence that when we're looking at the Phanerozoic and changes in ecospace through time, that this is probably fairly reliable. I'm very optimistic or cautiously optimistic about that. Of course, we need more studies to really agree, but this is very, very promising. And so because this is a Paleoverse talk, I wanted to talk a little more about sort of methods. I'm happy to answer questions about how to construct food webs as well. But I thought I would talk a little bit about these Malbachian cubes because there isn't actually an easy package where you can make these. These are all custom coded in R. And the workflow, they're quite difficult to make in R. The workflow is not simple or straightforward. And to my knowledge, I might be the only person that makes these in R. I think most people do it by hand. Um, so I just wanted to sort of share how you could try to do this in in R if you were trying to use this kind of approach, because again, it is fairly widespread in paleoecology. So here are our typical Bombachian cubes. These are cubes from another study that one of my former graduate students is working on. This is from the Ordovician. So Dr. Madeline S. created this set of cubes here. But again, we have our heat map, right? And we have our tiers and our three types of traits. So tiers, feeding mode, and motility. And so what we use is a package called Plot3D in R, and it has a 3D histogram function, and then also, of course, 3D text to go with that to put this text on here. And it's quite convoluted again, to make this because it isn't a straightforward, there isn't a package that's actually designed to do this by any stretch. So you sort of have to brute force it. The workflow that I've designed here or come up with, and I'm sure there's more efficient ways to do this as well. But essentially, you start with an empty data frame, which is telling it how many cubes we're going to have. Okay. And I do this for each tier separately. And actually, each one of these is its own figure by itself. So here we just have have one tier, and I believe it's the infaunal tier down here. And you can also see that I've kept the row names here as their own column, and that's because I'm going to need to merge these data with my actual observations. So these are all zeros, they're all empty. I'm just saying these are the cubes I'm going to want. And next, I take that empty cube and then my actual data. So you notice again here in the names for each row, and this is why they have to be there because they're here for my actual data. And you can see that in our actual data, here we had to convert from wide to long. And so when it does that, it put in these NAs. And so because we're merging these two and we have NAs versus zeros, it's just helpful to be able to do it this way. And so here are our actual data. We have actual richness for each of these functions. So we have four motile deposit feeders, for example, and we have to merge these two, and that gives us this data set here. So now we just have the column names, or sorry, the row names, and we've taken out that extra column because we only needed it to merge the data. And so now we have zeros for things that don't have any taxa in them. We have the actual numbers for things that do. And again, there are some columns here that don't exist because for this tier, we didn't have those traits. And so we've kept those in the merge, right? So we didn't have anything that was symbiotic in our data set. But that's one of our rows here, right? One of our sets of cubes. And so we carried that over and it shows as zeros. Then the other thing you have to do to fill the colors is to rank that richness, right? Because I want to use this heat map to show how many taxa are in a cube. And so we have to take that raw data and convert it to a rank. And this is the input you need for that hist3d function. You don't actually need the raw data to draw off the figure. What you need is the rank. And so you have to rank the data and substitute all of those numbers with their rank. And for us, rank of or zero, richness of zero becomes a rank of one. So you actually have to tell it, of course, that all these empty cubes are a color, right? In this case, light gray or white. And so that's color number one. So each of these ranks has to go with a color vector that we also have to create for each tier here. So for this data set, we have 1 through 4, so we have to have 4 colors to assign here. And again, these aren't the colors in order. These aren't these 3 colors. They're the colors that go with all of the ranks here in the scale bar, right? So there's a color for each of these ranks. And so there's actually a 0 here, right? Before 1, 1 is red. That's 2 in the data set. That makes sense. So it's actually quite complicated, but also easy in some ways, right? That's pretty straightforward. You just need your to convert your raw data to ranks and have it be the same number of cubes that you want, and then you make one for each of these, and you also, of course, have to make your scale bar. So I hope that's helpful. Again, a lot of people ask me how to make these. There isn't a straightforward package to do so. You really do have to write custom code to do this with R. But I think even just knowing about the hist3d function is helpful because it was quite challenging to find a package that did this. I will say that I made this code quite a while ago, so R changes pretty quickly, so there might be better packages out there to do this now. But again, to my knowledge, no one else that I've spoken to is doing is drafting these figures in R. They're usually doing it by hand. I've included the code here again just because it can be helpful for for trying to do things in R. This is the hist3d function, these are the sort of names for each of our sets of cubes. And again, here's our color vector, right? I named it C2 colors. C2 is one of the sequences that we're looking at here. And then here's the ranked data. So color var is actually the data input here. And then everything else that's required just to make one of those sets of cubes. And then I make the text labels separately. And if you notice here, we only make them for the bottom set of cubes. So we turn that off. We don't add them for any of the other sets of cubes because you only need them on the lowest tier when you're going to combine them in that kind of figure. But there's, of course, X and Y and axis. And there's axis labels for each of those. So I just included here the X as an idea of how to do that. And that's you add that on with the text3d function. And here add equals true is telling it to add it to after running this and creating your set of cubes, you're adding the text after the fact with this separate chunk of code here, and you would do that for each of your axes. So again, it's quite clunky. It's quite a lot. It can be difficult and challenging to figure out how to do this. So I just wanted to share a little bit about that. But again, I'm happy to answer questions about food web construction. I showed on some of my slide, we make our food web figures all using iGraph. So I'm happy to talk about iGraph as well. And we do our species level modeling for food webs in Julia. So we also have custom code that's published online for that from the workshop we taught at NAPC. But I'd like to, of course, thank everyone that's helped with all of this work, and NSF for funding our study on live dead fidelity. And I'll stop there, and hopefully I can be helpful and answer some questions.